An introduction to biotechnology. Today we will study about history and development of biotechnology. Biotechnology term is made up of bio and technology. Bio means biological system or processes, and technology means methods to create useful products. So biotechnology uses living organisms or cells to produce useful products. Biotechnology benefits mankind through various applications in medicines, agriculture, food, and environment. The term biotechnology was given by a Hungarian scientist named Karl Erike. Coming to the starting of the biotechnology is practiced since Paleolithic era. At that time, the domestication of the cattle and the cultivation of the crops like wheat and barley comes under it. The knowledge of the early fermentation processes like making of bread, cheese, wine, beer is also part of the biotechnology. Fermentation is a process in which microbial enzymatic conversion of organic compounds is there, and by the process of fermentation various products can be produced like idli, dahi, and various beverages are produced by the process of fermentation. Louis Pasteur proved that microbes cause the fermentation. By the process of industrial fermentation, various products like glycerol, acetone, butanol, and various acids are produced. Coming to the industrial biotechnology, the World War First and Second boosted the industrial fermentation. The discovery of the bioreactors allowed the fermentation on a larger scale, and it allows the mass production of penicillins and other antibiotics, hormones, enzymes, and pigments. Foundations of modern biotechnology. In 1590, Jensen invented the first compound microscope. In 1665, Robert Hooke observed cells. In 1676, Leeuwenhoek discovered microorganisms. In 1858, Virchow gave the theory that all cells arise from pre-existing cells. In 1896, Edward Buckner converted sugar to ethyl alcohol using yeast extract, showing that biochemical transformation can occur without the use of cells. Genetics and discovery of DNA. Mendel in 1857 gave the law of heredity using pea plant. In 1869, John Frederick Mischer discovered nuclein. In In 1882, Walter Fleming describes chromosomes. In 1952, Hershey and Chase identify DNA as genetic material, and in 1953, Watson and Crick proposes double helix model of DNA. In the recombinant DNA technology, a gene of interest is expressed in the DNA of the host cell, and then it is made to express the protein. So the steps involve enable cutting and joining of DNA fragments, creates recombinant DNA, inserted into host cell to express new traits, and it has wide applications in agriculture, medicine, and genomics. So applications of biotechnology includes in the area are there in the area of medicine for the synthesis of vaccine, insulin and gene therapy, in agriculture for GM crops, pest resistant crops, in food industry for fermentation and nutraceutical production, in environment for bioremediation and waste treatment. Some of the types of biotechnology are blue biotechnology, in which uh, application of the biotechnology for marine and freshwater organisms is studied, and the main motive is here to increase the seafood supply, to regulate the reproduction of dangerous waterborne organisms, and developing new drugs. In green biotechnology, the main motive is uh, to find out the environment friendly solution, and such as in plants to improve the nutritional quality, quantity and production of eco-friendly products. In red biotechnology, it is related to manufacturing of pharmaceutical products such as insulin, enzymes, antibiotics and vaccines. And the white biotechnology is related to the improvement in the industrial processes and other production processes. The use of enzymes as industrial catalyst is in eco-friendly manner for the production of valuable chemicals. So in the ancient biotechnology is related to the early knowledge of the food, shelter and domestication. The classical biotechnology is related to the fermentation based food production and medicines, and modern biotechnology is related to genetic engineering and RNA recombinant DNA technology, and biotechnology is applied in agriculture, crop improvement, food innovations, diagnostics, healthcare, animal husbandry, energy and environmental management. Different streams of biotechnology includes genetic engineering, where we study about the rDNA technology, protein engineering, bioinformatics, immunology, plant animal cell culture, biochemistry, cancer biology, environmental biology, marine biology, nanobiotechnology, biophysics, pharmacology and toxicity, cell biology, mi- microbiology, medical engineering, genetics. Thank you.